morning, everyone. Welcome to today's Strategic Farming Let's Talk Crops program. So we're glad you joined us for today's session on biological control or using other organisms to essentially control pests for us. These sessions are brought to you by University of Minnesota Extension and generous support from the Minnesota Soybean Research and Promotion Council and the Minnesota Corn Research and Promotion Council. I'm Anthony Hansen, Extension Educator in Integrated Pest Management based out of Morris. Today, we're welcoming Dr. George Heimpel. He's a professor of entomology and the Department of Entomology who focuses on biological control, especially dealing with beneficial insects. This discussion will focus on questions that we received at registration, and also a reminder for future sessions that we do have that option where people can send in questions ahead of time. So each session is recorded and it will be posted at a later date on our website. With that, I think I will turn it over to George. George, thank you for joining us today.
Thank you very much, Anthony, for giving me the opportunity to speak with you all this morning. It is truly a pleasure. And I'll just read the topic of my presentation, which is biological control of soybean aphid, the parasitoid Aphelinus certus. I'm going to assume that most of you know about soybean aphid and know that it invaded the United States in the year 2000. And what you may not know is that shortly after that, in 2001, myself and others went to the native range of soybean aphid, which is China or Eastern Asia more generally, and we looked for tiny little parasitic wasps, otherwise known as parasitoids, that were attacking soybean aphid back there in its native home range. And the point of that was to discover whether there were any possible biological control agents from the native range that we could bring over and release in our soybean fields. And so that was a long process. I think Anthony was kind of around when some of that stuff was going on. And this is just a little map and some pictures and some names of some of the parasitic wasp species that we found. So they kind of came from various parts of the native range of soybean aphid, which includes basically Eastern China, South Korea, and Japan were some of the places that myself and others went. And here are some of the names and pictures of some of the sort of more important species that we found. The total number of species, which includes some things that might not have actually been species, but just strains of species, was about 20. Some of these went through sort of extensive safety testing within our quarantine lab at the University of Minnesota. I think maybe some of you have gone to take tours of that site. And out of that process, there were two especially that we found were sort of highly specialized for soybean aphids and close relatives of soybean aphids. And so they got sort of the governmental stamp of approval that we can release them. And those two main ones, one is called Biocontrol communis up here, pictured here, and another one is called Aphelinus glycinus, pictured down here. The other ones tended to be sort of more broad generalists, meaning that they attacked a lot of aphids, which to some people might think like, well, that doesn't really matter that much. But the problem is that there are native aphids out there in nature, and we're sort of not supposed to bring in natural enemies that can attack native species. And so we could only bring in those ones that were highly specialized, these two. Now, we spent a lot of time, and I had a few graduate students working on this, not only in the lab trying to determine whether they'd be safe, but then also in the field, releasing them and seeing whether they would have an impact. And I'm really sorry to say that neither of those two species were able to establish in Minnesota. And I could spend this whole time talking about why not, or about theories about why not. But I'm going to skip over all that because that's a long time ago and they did not establish, and we have to move on. And then, and by move on, what I mean is something happened, and that's that one of these species. I'm going to kind of go back and forth here. You know, you'll notice this name here, Aphelinus certus, which was one of the ones that we found in China and that we tested and that we couldn't go forward with because it had a broad host range. It attacked a bunch of different aphid species. So it was not one that we even sought a permit for, but lo and behold, it came on its own. So
St.
basically,
George,
yeah.
you want to mention a little bit about the facility on campus for isolating insects or quarantine, kind of what goes on there? Because I think a lot of folks maybe haven't seen or heard of that potentially outside of the biological control realm or invasive species.
So, Anthony, I didn't quite hear your question in its entirety, but I think you were asking me to talk a little bit about the quarantine lab in general.
Yep,
Is that
exactly.
right? Yeah. Okay. So the quarantine lab is a structure that is on the St. Paul campus of the University of Minnesota, but it's actually owned in part by the Minnesota Department of Ag. And it's sort of administered partly by the University of Minnesota and mainly actually by the Minnesota Department of Ag. And the point of a quarantine lab, this is technically called a BSL-2. There's also a BSL-3, which is attached to it, but we were working in the BSL-2, which stands for Biosafety Level 2, which means it is meant to contain insects. So if you work with an insect inside this lab, you can be pretty sure based on how the lab was built and how the airflow is managed in the lab and the protocols that we have for working in the lab, that insect is not going to escape out into the world. And the reason that we need that kind of a lab is that we want to study these non-native insects that might be beneficial, but we can't bring them into our own lab, which has, well, in our case, it doesn't have windows, but it could. And, but, it has doors. So, the insects could easily kind of fly out of a cage, or they're just sort of out in the space of the lab, and then they could get out, and then they could get out and, escape and, establish. So a quarantine lab is where we can safely do the kinds of studies that are needed to evaluate biological control agents. Is that kind of what you were looking for, Anthony?
Yep.
Did you want to add anything?
That's great, George. Yeah, I was just thinking mostly just a little plug for the quarantine facility because I know that was something new to me even 10 years ago. It's kind of a cool facility how it's set up. So yep, you described pretty much exactly the key points up there.
And our quarantine lab opened in 2003. And this project, as I mentioned, started with the first trips to China were in 2001. So basically what ended up happening is that our project was the first one to sort of occupy that lab, or like the one of the first two. There were two at the same time. So yeah. It was, if we hadn't had that lab, then we would not have been able to do that previous work that I mentioned. But the thing about this Aphelinus certus is that even though we tested it in the lab, we never wanted to introduce it. And then what I want to talk about during the rest of this presentation is that it came on its own. So here I'm saying that it's native to Asia, we know that. And then it became accidentally introduced first on the East Coast by like 2005. It was first found in Pennsylvania, and it got to Minnesota by 2011. So it came on so. And by now, and pretty quickly after 2011, it became widespread in Minnesota, and it's basically, through all of our weather events and through all of our years of relatively low aphid densities, it has been persistent. So these maps kind of show you some of that with these blobs basically telling you how many of these parasitic wasps per plant were found. And the little dots just showing where we looked over all of these years. And so that's sort of the first point to make, is that this little wasp is now sort of an integral and an important part of the natural enemy fauna, meaning those insects that attack soybean aphid in Minnesota and also in a few other states. And, again, kind of thinking about, the timeline here, I have a graph that goes from 2011 to 2021, and it has two lines on it. One of them talks about the soybean aphid density over that time. This is showing what we call the cumulative aphid days, which kind of gives you an idea of basically the numbers of soybean aphid over the whole season. So, the higher this number, the more there are. And this line basically trends downward, and significantly so. So basically showing that over the last 10 years, the density of soybean aphids has gone down. And I think most of you that work with soybean have probably experienced this, and that's that the last few years, the last 5 years or so, have been relatively low aphid years in at least most parts of the state. At the same time, this blue line is showing what's happening with our little parasitic wasp. So, remember that it came in by 2011, it might have been here a bit earlier. And over the same time that the number of soybean aphids has gone down, the rate of parasitism, that's what's shown over here, of this little wasp on soybean aphids has gone up. So just to show you a little bit about what this axis means, this is what we call the Aphlinus fraction. And it basically goes from 0.01 to 0.1, which basically means from 1% to 10%. So even this high number, it doesn't seem high maybe to many of you, but 10%, as you'll see a little bit later, can actually do a lot. So basically, though, the take-home message from this graph is that over the last 10 years or so, soybean aphids have gone down, while the parasitism rate on soybean aphids by the sow wasp have gone up. So there's kind of an obvious question, and that's, is Aphelinus responsible for the observed soybean aphid declines? It kind of looks like it. I mean, the trends are sort of in the right direction. And so that's something that we've been trying to figure out over the last years. And I'm just going to kind of go over some of the investigations that we've done to get at this. First thing is that I had a student, Joe Kaiser, who I'm sure Anthony remembers Joe, in our lab. And he did this experiment in about 2015. It was then published later, in which he basically did use various cages to either exclude the natural enemies or not. And he had a certain kind of mesh that would exclude the predatory insects but leave the wasps inside. And so if you look at this graph, showing the number of soybean aphids per plant after a certain period, basically from when the experiment started. So, I have numbers going up to about 500. And in this treatment here, this is a treatment where there were cages put onto the plant such that no enemies could come in. So no little wasps, no lady beetles, no lacewings, nothing. And in that case, you got about 500 aphids per plant in this, experiment. And in all of these other treatments, the wasps were, allowed to enter in various different ways. This treatment is kind of the most important because it's the one that was allowing only these little wasps to get in, no other insects like ladybugs and that kind of thing. And you can see that there's a huge difference between the number of aphids when the wasps were excluded versus when they were included, when they were allowed, just naturally, just what's out there in the field. And, some of you might know too, this happens to these densities of aphids per plant happen to be on both sides of the economic threshold, which is 250 aphids per plant is when the farmer would spray, or when we would suggest that they spray. And so, it just happened to be in this experiment that the wasps were able to keep the aphids, the wasps alone were able to keep these aphids down below that spray threshold, which is really kind of nice. So it was suggesting that these wasps were, effective enough so that farmers would no longer need to spray against soybean aphid, which was very exciting for us. And it certainly happened in this field. But, other people were looking at this and then we looked at it later. So let's see what else has been found out about the system. So, around this same time, there was a lab in Quebec where they also have soybean aphid and they also have Aphelinus, certus. And they were looking at it too, and they did their experiments a little bit differently than we did. They used more of sort of a mathematical modeling approach, and they came up with this graph and this kind of an analysis showing that this kind of a trajectory is what you would expect soybean aphid to do by itself with no Aphelinus in the system. And this tiny little dotted line, or it's not a tiny line, but it's not very distant from the previous line, is the one where the wasps were having their effect. And you can see that they basically had very little effect here. And so while we were saying, oh, this thing is great, it can lower the densities below the spray threshold, these other guys were saying, well, in Canada, maybe not, So it's looking kind of muddy. I mean, I think what just this comparison shows is that yes, they can do this at some sites, some years, but this is saying that they don't necessarily always do it. And so anyway, after Joe was done, they had another student. They kind of wanted to look at this. His name was James Mixanic, shown here, setting up some of the same types of cages that Joe had previously used, and probably Anthony remembers James, or maybe James is a little bit after your time.
Anthony? I think I was in grad school at the time for both folks there. So
okay, good.
This does remind me, George, you mentioned lady beetles briefly, and we don't need to go in depth into it. But one question I do get sometimes is people ask about the Asian lady beetle, the one that invades into our homes in the fall, and they're wondering if that was introduced to control soybean aphid. We know it works well for soybean aphid, but you want to just really briefly mention what?
It's the Asian lady. Yeah, so the Asian lady beetle, this is kind of a myth because the Asian lady beetle, yes, it is an important natural enemy of soybean aphids in our fields, yes. But we did not purposely release it against the soybean aphid. It came on its own, much like this Aphelinus certus, it just came on its own. There is some truth to the story in the sense that this beetle had been previously released in southern states like Georgia to control other aphids, and it might be that those populations then moved their way, northwest to us. That's one possible route that they could have gotten here from. But anyway, if anybody tells you that any entomologist in Minnesota released those beetles against soybean aphid, you can now tell them that that's not true. Anyway, thank you for that question, Anthony. I have heard that too. Okay, so this is James in the field, and this shows a little bit more the kinds of things that James did. So this is actually similar to what Joe did. There were different kinds of cages. There were some cages that had a kind of a broad mesh, so that the wasps could get in. The wasps were tiny, so I maybe mention this, but they're literally the size of a period printed on a page. They're so tiny, and so they could easily get through a kind of a mesh like mosquito netting, but that would exclude the larger predatory insects like the lady beetles and the lacewings and those sorts of things. So some of the cages were that type. Some of the cages, though, have such a fine mesh that they would even impede our little wasps. And so James set this up in various parts of the state. He had these really pretty kind of pictures from the field. And rather than Joe, who just did one really cool study at one site, he did it over a few years and at different sites. And so these are the results that James came up with. And so not quite as pretty as what Joe found. So here, this is kind of a complicated figure, and I don't even want to go through all of it, just to show you that here what we're looking at is the daily population growth rate. So basically, if it's at 1, for this number, it means that populations didn't change and they stayed the same. And out of these 3 treatments, the one in the middle is always the one from which includes the wasp, but not the predator. So what you're looking is basically for this number to be the lowest, which it was, trending in some cases, or trending to be lower than total exclusion. But as you can see, there's no super strong evidence of the kind of control that Joe saw. And so basically, if you take all these data together, you say that, well, in these fields, in these settings, there wasn't strong control of soybean aphid by this little wasp over the time frame that the experiment was done. Doesn't mean that it didn't happen at all, but we don't have strong evidence for it. There's sort of another way to look at this. Wait a minute, here we go. And that's to sort of try to ask what was the reason. And one thing that we can do is we can look at this sort of population growth rate. Remember that 1 is where you have even growth. And then we can look at the percent mummies. And I'm sorry, mummy is kind of a jargony term. It basically means the pupal stage of the wasp, which looks kind of like a blown up black aphid. And so we can count these on the plant. And so this is basically saying, for example, there were 2% of the aphids that were this kind of parasite mummy. And what this graph is showing is that, yes, the higher the parasitism rate is, the lower the aphid growth rate is, which is good, but it doesn't get high enough because we don't get down to this magic number 1, which is even growth. Of course, if you go below 1, that means that the aphids are declining. And if you think about this graph a little bit more, you could kind of extrapolate it out. If you assume it keeps being linear, which these tend to do, then you can see that it would take about 8% mummies. So 8% of the aphids being parasitized to get to a population growth of 1, which is staying stable. Of course, if you get more than 8, then the aphids would be actually going down in size, in population size. And so what this shows is that, yes, these wasps can lead to lower population growth of the aphids, but we just need more of them. We need more of them because this is only going up to about 3% and we need about 8%. And so we went a little farther with that by doing some so-called mathematical modeling, which is when you kind of take everything that you know about these insects and about the conditions in which they live, and you kind of plug it all into a bunch of equations, and that lets you predict what might happen in the field. And this kind of talks about what kind of model it was. It's called a matrix model, and it had stage classified coupled equations. It just means that we looked at like the young aphids and the old aphids, and then we basically tried to simulate it over one field season so that we could connect it to what's going on in the field. And another thing is that we had a certain sort of parameter in it saying that if there's no parasitism at all, if the aphids are not controlled at all by the parasites, or just if the parasites aren't there, you'd get 6,000 aphids per plant. And let me just show you the kind of things that we get out. So you get out these kind of graphs, which show sort of the population density of the aphids here in this line, and then the parasitoids in this line. And the first thing I'd like you to notice is that the maximum here is about 2,000. And remember that without any parasites, it'd be a 6,000. And and I didn't mention that this gray part just kind of shows the variability depending on how you sort of change some of the aspects of that model. And but this plot is showing sort of what you would expect under the most normal of conditions. And you would expect the aphids to grow to a density of about 1,000 per plant, which does cause economic damage. But then this gray stuff is saying that it doesn't always reach that level, and it can also reach more. And one way to look at this is to think about the economic injury level, which under most conditions is about 700 aphids or so. So if you have 700 aphids per plant, there's going to be a drop in yield under those conditions. And so if you look at sort of what part of this gray part could happen, it's what this is saying is that in 31% of the times, another way to think about it is in 31% of the fields, you would have suppression of soybean aphid below the economic injury level. And another number to think about that you probably know about is the economic threshold, which is the number of aphids above which a farmer would be advised to spray in order to save money. And so that's 250. And by the same reasoning, based on this gray area, what percent of the model runs ended up with aphid levels below the economic threshold? It was about 10%. So you could look at it as in about 10% of the fields, you would expect this little wasp to drive soybean aphid levels below the economic threshold, which means that a farmer wouldn't have to spray. So that kind of jives with the fact that Joe Kaiser, my first student, found an effect. James, my second student, found sort of variable levels of effect, but not very strong. So it kind of makes sense that yes, they can control soybean aphid, but they don't always do so.
George, I think that's a good point to put a little plug in again for the thresholds. Just a reminder that 250 aphids, that's something most folks know. That's actually, as a reminder, not the point where damage is actually occurring or significant damage. It's that near 650, 700 level that we're actually seeing that break even point. So we always like to remind folks that even though we teach people 250, that's just giving you that week's lead time till there's actual damage.
Right.
George,
Right.
I think we got about maybe 5 minutes left, something like that.
Okay. Okay. Well, I think yeah. So I guess I'll go I'm just going to kind of skip this whole part and just, I'd like to talk about sort of the broader life cycle of these wasps, and especially when they tend to emerge at the beginning of the season, so in the spring, and how we might be able to improve their ability to emerge. So this graph right here shows when they tend to emerge from the field if they're overwintering within an actual field. So let's say you have a soybean field and you have these little wasps and they drop down into the leaf litter of the field. And then what happens the next year when it's actually a cornfield? At what point would they actually emerge out of the soil? It would be at around the 10th of June, which is actually a great time. This is a graph that shows it. It's a great time, so that's good. But we don't exactly know where they tend to overwinter. They might also overwinter in woodlots because the overwintering host of buckthorn, of soybean aphid is buckthorn. And if they're there, if they're overwintering in a woodlot, then they emerge much later. And this graph would show you that this would be too late for them to have an effect. And so we want to help them to overwinter within soybean fields as best they can. We actually do think that this is mainly where they tend to overwinter. And so what we wanted to do was to look at the effect of tillage on the ability of these little guys to overwinter. And and we tried a bunch of studies, it was actually hard to do. So then we ended up doing a really simple thing where we just basically put a bunch of mummies in these little bags, either just onto the ground, or we bury them with only 4 centimeters of loose soil to kind of simulate what might happen under tillage. And then what we found, as you might expect, is that if they're placed on the top of the soil, they do a great job emerging. If they're placed under just a little bit of soil, they actually cannot emerge at all, and they basically die. And so this makes us think that probably a no-till kind of a cultivation is going to be better for Aphelinus certus. It's going to allow them to overwinter in fields and to emerge in time to suppress soybean aphid. And I think with that, I can just do the conclusions. I think Anthony was kind of suggesting that I don't have that much time. So I think I've already said all those things. And I think I'd rather just have time for questions and go to my last acknowledgement slide. So I'd like to thank my students and our funding agencies. And I think with that, if there are any questions, I would be glad.
Yeah, we do have a few questions coming in, George. So
Okay,
again, thank you for covering the good summary of things there. And that's just a little snippet of what goes on with biological control, really just for one parasitoid here. Obviously, there's a lot going on with other species, beneficials, other pests. So it's a pretty diverse area. So we do like to put a plug in for biological control. That's a major component of our integrated pest management systems. Like we said, for soybean aphid, there is control out there. So that's basically a free benefit in a way for growers, especially when we are looking at our insecticides. If it's less broad spectrum, then we might get both our insecticide control and keeping some of our biological controls out there too. So that's why we focus on IPM in different areas. So George, I'm going to take a couple questions here, folks. One, this is Susan just asking, basically, so what you're trying to summarize, I think that graph you had where it extrapolated out to 8% mummies or infestation. She's just basically asking is you need more parasitic wasps to effectively control soybean aphid?
Yeah, I guess that is basically what we found there, is that they are around and they can have an effect on aphid growth, but they're just not around in numbers that are high enough at least in those fields, in other fields, they are around in higher numbers. I actually think that this overwintering stuff that I was mentioning is probably an important bottleneck that they have. I think that a lot of them either are not able to overwinter, so they just die over the winter, or they are trying to overwinter in areas where they're going to emerge like too late to really have an effect. Or one thing that we don't really know about is the interaction between soybeans and corn. So, of course, we tend to rotate soybeans and corn. So, if you have an Aphelinus certus that's overwintering within a soybean field, which, as I mentioned, is where we think most of them do try to overwinter, in most cases, they're going to emerge into a cornfield, right? And so that's a bit of a black box for us. We don't exactly know how well they would do in the spring in finding a soybean field from a cornfield. I expect that if they're on the edge of that cornfield and they're right near the soybean field, it might be pretty easy, but what if they're in the middle? So I think that might be another reason that the numbers aren't quite as high as we'd like them to be. But I think in either case, whether you're talking about soybean to corn or even the rarer soybean to soybean, we do think that a no-till or a low-till system would conserve them better than a complete tillage system.
So George, I got a couple of questions coming in, so I'll kind of lump them together. These are asking more about the insecticide tolerance of these parasitoids. And one of the specific ones that was being asked is, what about any prophylactic applications of insecticide? How is that affecting parasitoids, especially if it's, say, R3 to R5? And if we're getting more situations like that where people are putting them out without necessarily doing the scouting and things like that, how can that disturb the system pretty much there?
Yeah, I mean, that is a good question. And so we actually had some colleagues come in from Spain and do exactly that experiment, in our fields. And so they checked whether aphids feeding on seed-treated beans. So this is, seed treatment includes, the neonicotinoid insecticide thiamethoxam, whether aphids feeding on those and then parasitoids attacking those aphids, whether there would be a negative effect on that. And what they found was that, okay, just a little bit of background, and that's that when aphids feed on plants, they have to process the plant juices, and they excrete a very sugary substance that we affectionately call honeydew. And if you go out into sort of an aphid encrusted field, you can tell because it's all sticky, and that stickiness is honeydew. And the thing about honeydew is that a lot of insects like to feed on it, including parasitic wasps and other beneficial insects. And what our Spanish colleagues found in our field, so this is relevant to Minnesota, is that when these parasitic wasps feed on the honeydew that was produced by aphids feeding on seed-treated plants, then it can negatively affect them. So the honeydew is basically a pathway for the insecticides to get out into the world, and so then they can be eaten by the wasps. Now, the questioner was really smart in asking about the stage of, the soybean plant. And, it is true that once you get into the later stages, the insecticide is basically worn off. And so at that point, it doesn't have much of an effect anymore, neither on the aphid nor on the beneficial insects feeding on the honeydew made by the aphids. But there's sort of a window of time when they are toxic, and that window of time is unfortunately kind of right when you do need the wasps to start to build up and to be able to sting aphids in that sort of early July period. And so it is true that that particular interaction could also be having a negative effect on the ability of these wasps to control the aphids.
How about the foliar insecticides? I imagine something like a broad spectrum pyrethroid, probably they won't have much, if any, tolerance to. Has there been much look at some of these other foliar insecticides?
Yeah, there were some early studies on I can't remember the name.
Probably lambda-cyhalothrin.
Yeah,
one of the pyrethroids. So
I know, yeah. That one, and I think some of the others, and yeah, definitely highly susceptible. And those also have the effect, to the extent that they're effective against aphids, they're also going to leave the wasps with no aphids to attack. So they would, yeah, so definitely spraying broad spectrum or really any foliar insecticide, I think, is going to have a bad effect on this biocontrol interaction for sure. Yeah.
Yeah, I can't say I've seen literature. This would be a Bob Cook question if he's had any studies on it, because I know they've looked at some of the newer chemistries and they're a little more targeted towards aphids and say things like lady beetles aren't affected as much, but I can't recall seeing parasitoids in those studies yet. Sure,
Yeah,
I gotta think it's out there, but I just haven't seen them yet.
He actually had a student in his lab that was looking at that for these wasps, and I guess the data are still being analyzed or something, because I haven't heard what the outcome is. But yeah, I mean, I guess it's true. That's a fair point, that there might be some more specialized insecticides that wouldn't directly kill the wasp. In a lot of those cases, there would still be like some sublethal effects, and then there's always the issue that of course, it's great to kill off the aphids, but if you're killing off the aphids, then the wasps don't have anything to control. To attack. So you might be like, in the short run, that might be great. You have lower aphids. But it could also, in the longer run, it could just kind of depress the population of wasps,
Yeah, I think that's a good example of when we deal with insect flare-ups, a lot of times it's something happened to the natural enemies, whether insecticides or they just kind of got a mismatch and didn't have enough hosts available. And all of a sudden those insects or the pests are kind of released from their predators. They take off. I think I got a couple more questions here, so I might as well keep those going. One, Larry's asking about if there's any more information on the prevalence or effectiveness of the parasitic wasps from China. I'm assuming that maybe the studies that were done a while ago, just kind of what the background is on where people would go to learn more about what was done there.
Where they would go to find out the information on those. Well, they could go to the scientific literature for sure. There's a lot of that. So I guess I would have to point them to that. I think way back in the day, there was some sort of pamphlets written up too. But basically, it didn't, they didn't establish, so there weren't that many sort of, extension bulletins written about it, in the sense that it didn't work. We have some theories for why it didn't work. Some of those theories have to do with sort of, the kind of crop that soybean is, where it's an annual crop. So it's not really a stable ecosystem for the wasps. And then, the aphids overwinter on a different host. So it can be kind of hard for a specialist natural enemy to be able to establish under those conditions. But anyway, that's sort of a very long conversation to get into that.
Yeah, I can remember some classes I took from you, George. That was, I feel like, weeks of dealing with that topic there. Next question I got here is just asking, has anyone been looking in the adjacent states of Minnesota? Do you have a snapshot or idea what these wasps are looking like in, say, Wisconsin, Iowa, or the Dakotas?
Oh, yeah. So we are working with people in Wisconsin and Iowa, as you mentioned, and also, well, with the with funding from the NCSRP, we've been taking a more regional approach. And so these wasps are present in those other states. Some of those other states have been having even lower aphid years than we've been having. So that makes it kind of hard to get data on the wasps, when the aphid densities are so low. But they are, they have definitely been found in Iowa, Wisconsin, North Dakota, South Dakota, Ohio, Michigan, Missouri. So they've been found throughout the whole region.
All right, I think last question it looks like here. This question is asking, do the Asian lady beetles control soybean aphid better than the parasitic wasps right now? Probably a complicated question a little bit, but
I think that's a good question. I think that the Asian lady beetles can. I think that the Asian lady beetles are better at kind of coming in and controlling when there's already a lot of aphids. So, kind of like the big thing about the Asian lady beetles is whether they go through, just kind of come through fields and kind of pick off a few aphids, or if they kind of settle down and lay eggs and go through a whole generation and that kind of thing. And when the aphids are at low levels, they just kind of come through, they don't really lay their eggs. When the aphids are at high densities, then they'll do that kind of stuff, and they will be able to control aphids, but they don't tend to control them that often below the economic threshold. What they'll do is that they'll knock them down by the end of the season so that the following season there aren't as many. It's the kind of thing that they tend to do. So I think the answer is they probably are more important than Aphelinus at this point, but I think that Aphelinus is probably better at keeping the aphids low if they can get into fields relatively early and kind of stop things from an early point in the season.
Yeah, so kind of a good network of beneficial insects when you look at it that way. All right, well, thank you again, George, for joining us today. And otherwise, thanks again to our sponsors, Minnesota Soybean Research and Promotional Council and the Minnesota Corn Research and Promotional Council. That's definitely helped out with this program in terms of what we've been doing here and then in previous iterations of strategic farming as well. Just for a reminder, next week's session, and I believe it's our last session, if I looked at the calendar correctly, it is managing soil fertility and weed management costs. So obviously we talked about supply chain issues and everything happening with soil or with fertilizer prices and also some of our herbicides. That's coming up quite a bit. So that should be a good session coming up to wrap things up. Otherwise, have a great rest of the day and we'll see you next week. Thank you, everyone.